electricity, so I think, is there a clock back there? Yeah.
Okay. All right, we'll go ahead and start. I think people will find their way. For our last talk for the morning, I'd like to introduce Robert Penner. Again, this is a very different talk from the first two, but the theme is actually very interesting to try and unite in your brain. It's actually, I wouldn't want to insult the reader here. You can make the dots connected if you do. It's very interesting how these are connected. But in any case, his connections in the world have, up until fairly recently, for some reason, I was under the impression it was Caltech, but it seems to be sort of everywhere. Other than that, I'm not exactly sure what happened. But there's he has affiliations at Aarhus and at the Max Planck Institute in Bonn at the University of Tokyo, and so he's like an electron. But his primary place in the world is at the Institut des Études Scientifiques in Paris. It's a very interesting place. If you you might want to take a look at that. If you don't know what it is, it's an amazing place. It's a place that a very special place in the world. But and it's had some incredible people in it, and one of them was René, Thom, and apparently he left his chair, and Robert seems to be sitting in it. And so yeah, so he's a mathematician. Obviously, his main interest, I guess, is I'm reading here, or geometry, which is kind of interesting, but and algebraic number theory, string theory. But more importantly, he's interested in biology and in the mathematical underpinnings, I guess, of understanding life. And fundamental to that is the you know, if you will, spontaneous appearance of structure. And so that needed a theory. And so the theory of morphogenesis is something that he's developed and worked on, and he's going to tell us about it today. Thank you.
Thank you, Chris. So let me thank Christian and Chris for the invitation here and the opportunity to speak with you. Indeed, I'll speak about morphogenesis, which is literally the birth of form. And this comes in, I should say, this is joint work with Andrei Minarsky and Nadja Morozova and Christophe Soulet, a physicist, a biologist, and a number theorist, and I, a mutt of some sort. Right, so morphogenesis, the birth of shape or the birth of form, maybe the key manifestations being, say, embryogenesis. Embryogenesis. So how does the zygote develop into the adult organism? Where is the data stored? How is the information coded? So first of all, embryogenesis. There are hundreds of experiments in the literature involving transplantation and regeneration. Transplantation. I'm just trying to give some of the manifestations of morphogenesis in which we shall be interested. Transplantation, regeneration, which we'll discuss at the end. Regeneration. And maybe another extreme example of this is morphogenesis gone wrong in the form of carcinogenesis. So there are various manifestations carcinogenesis of this birth of form. My goal here is to describe in several, specifically 7 explicit kind of axioms, a model that explains many of these phenomena in these several manifestations. And in fact, the model I propose is intended to be, and indeed is, explicit enough to be implemented on the computer. So we have been doing, we've been probing the model with explicit computer simulations. And again, I'll come to that later. But this is the goal. This will be one of these talks where I'll end with a list of axioms, and I'll introduce and discuss them as we go along. So it's sort of, maybe I'll try and use the rear board for the axioms. So let me first of all start off with mathematical biological ansatz as follows. Let me say, morphogenesis, so the shape of an organism is definitely a multi-scale problem. For various of the information depends on the cellular level, so the micro level, whereas on the other hand, you want to distinguish an elephant from a giraffe from a zebrafish and so on, that's obviously large scale. So this problem is inherently multi-scale. And let me start on the microscale, and let me just discuss a cell and introduce a mathematical biological principle that I will rely on in formulating the model. So what does a cell look like? So there's some cell here like this. And the ansatz is that a cell is what's called star convex, with respect to the centrosome, or the centrosome, AKA the MTOC, the microtubule organizing center. So there's the nucleus of the cell, and just outside that is this sort of pair of cylinders. I'm not drawing things quite to scale, of course. Is this pair of cylinders called the centrosome. And the centrosome emanates a collection of gamma tubulin tubes that hold the cell surface in place, much as a circus tent is held by a bunch of poles. The tubules coming from the MTOC hold the cell surface in place. And the notion of star convex is a notion in mathematics that says a set in space, again, this is meant to be 3D, a set in space is star convex if with respect to a point P, if for any point in the set, the line segment from the specific point to that point is contained in the set. And my ansatz is that cells are star convex with respect to the centrosome. As I say, with this circus tent analogy in mind, that's a reasonable biological ansatz, and it's a very useful mathematical ansatz for us here. There's axiom 0, which I won't bother to write down, is there is no statement in biology that is always true except this one. So you can, I'm sure, for instance, a nerve cell you will see is really not star convex. Actually, it is nearly so, but is not star convex. So everything has to be taken sort of with a grain of salt. So a consequence of star convexity, remember I wanted to talk about micro shape, morphogenesis on the micro level. And this ansatz allows us to describe the shape of a cell by a real valued function on the two-sphere. So imagine a small two-dimensional sphere here. And here's the centrosome. And let us look at the cell boundary. And here is some vector x, a unit vector x, or some small vector. You normalize the two-sphere to some size. And then the cell boundary is given by some, let us call it, rho of x times vector x. Rho of vector x times x. Where rho is a function from the two-sphere to the reals. So this is already a use. I'm showing you the utility of this ansatz, which will occur again. Is various biological quantities can be described by real valued functions on the two-sphere. In particular, the shape of the cell. So in some sense, we've satisfactorily described at least the morpho part of morphogenesis on this small scale. So let me come to the, hang on one sec. Let me come to the first axiom. And like I say, I'm going to try and keep the axioms back here. So to dignify them, I'll use Roman numerals. So the first axiom is that each cell comes equipped with a cell surface distribution of compounds. I'll be more explicit in a second. Cell surface distribution of compounds. And I'll refer to this as the epigenetic, which I'll often drop, spectrum. So a cell comes with a spectrum which describes a cell surface coating of compounds. To be explicit, well, first of all, epigenetic, let me discuss that term. Whatever is driving morphogenesis, it is beyond the genome, literally epigenetic. For all of the cells in the organism share the same genetic material, of course, and yet the cell fates, as you vary over the organism, are very different. So whatever it is that is driving morphogenesis is epigenetic. And this first basic concept, actually my colleague Nadia Morozova introduced this notion some years ago, that somehow morphogenesis is driven by these cell surface compounds. To be explicit, she and we shall imagine a cell surface coating of oligoconjugates. So you can imagine some in fact, cell surfaces do indeed come equipped with a very complicated distribution of oligos, which you might think of as a word of length, say, 6 to 12 a word of length 6 to 12 in the monosaccharides. So I'm trying to argue that there is plenty of combinatorial complexity here by the time you're looking at words of even some reasonable length in monosaccharides. All right? The model that I will describe and the axioms aren't really married to this idea of using oligos as the cell surface coating. So if you can imagine proteins or other compounds as well as part of this cell surface code. And now let me observe that this, maybe I'll come back here, that this star convexity is likewise useful to describe this distribution, the cell surface distribution, because likewise, to the manner in which we describe the shape of the cell, a cell surface distribution of whatever, oligos or whatever you want, distribution is also described by a real valued function on the two-sphere. Is also described by so there will be some collection of compounds, F sub I indexing the various compounds we're going to keep track of. So some collection of functions on the two-sphere real value that are simply giving the concentration or whatever the density, some measure of how much of compound I is nearby. So a density. I maybe it's best to think of this as a density. And again, where I goes from 1 to the number of species we are going to keep track of. Cell surface species we're going to keep track of. So there is our first axiom. And a way, again, remember, I'm trying to describe something that can be implemented on the computer, and that's why I'm being sort of practical and explaining, these cell surface distributions can be thought of as functions on the two-sphere. Okay, we come already to the second axiom, which is that each cell sends a signal. Well, let me transmit or sends its spectrum, its epigenetic spectrum, to a collection of neighboring cells, and again, maybe I put this in quotes for the moment and then explain neighboring cells via what we shall call, I'm using sort of the mathematical convenience of when I'm defining a concept, I underline it. So the fact that I underline this means that's what I mean. When I use the term spectrum, that's what I mean. And likewise, when I use the term cell signaling, since I've underlined it, this is the definition of that concept. So it is, many experiments in the literature show that there is that cell-to-cell communication drives or mediates morphogenesis, and this is our second axiom. And I'm making sort of the simplifying assumption that the cell transmits its spectrum. It is in the interpretation of that spectrum that distinctions can be made. But the cell it doesn't have some special code. It just says, I am I, in the sense of transmitting its spectrum. Okay, so that is the second axiom, and second and critical axiom. Next, we have the notion of cell events. Each cell at a given instant, performs one of several one of several cell events. And again, I underline this, and so I'm defining, this is the notion of cell event. And there's a finite list of possible cell events. First of all, there might be a change of its epigenetic spectrum. This is sort of an internal cell event where the cell changes its spectrum and hence its signaling. Change of spectrum. It could move. A cell could move. It could change shape. Change of shape, namely change of this function rho. Change of shape. It may divide, producing two daughter cells, and it might also die, apoptosis, programmed cell death, apoptosis. And within this model, those are the various possibilities. That's what a cell can do. Now, on what basis does it make? Let me make sure I didn't forget. Yeah, right, apoptosis. On the basis of what information and in what context does the cell choose which cell event to perform? And that's the next axiom, is that the laws are universal throughout nature and common from one species to another. So at this point, I'm arguing that somehow there is basic physics involved, and that should be universal across nature. So, and then we'll draw some consequences already from these first four axioms. So the fourth axiom is there are, and again, I'll be very explicit here, universal rules by which, more explicit, universal rules obeyed throughout nature, and I'll capitalize nature, obeyed throughout nature of the cell event, and now here I be more explicit, these universal rules and the determination of cell event depend upon the cell's spectrum and the received signal. So of cell event, which are determined by the cell's spectrum. I realize I forgot to say something. Let me go back and fill in a gap. By the cell's spectrum and received signal. And remember, I put neighbor in quotes over there, received signal. I put neighboring in quotes over here. So in what sense, to whom am I going to send my message? it seems clear I would send it to my immediate spatial neighbors, but I mean something much more than that. There are long-range chemical interactions, so neighbor doesn't mean neighboring in space, and indeed these could be ligand, ligand receptor, ion exchange. I'm imagining neighboring to mean anything, any of the possible biological pathways that you could think of by which two cells could communicate, not neighboring in any spatial sense. So now let me already formulate a consequence of these first four axioms and a concept that will come later when we discuss regeneration and transplantation, namely the notion of harmony. So a cell is sitting here, and it's got its spectrum, and it is receiving this collection of signals from neighbors, possibly not proximate. And in the adult organism, in this fully differentiated organism, the cell is told equilibrium. Sit there, do nothing, you're an adult, sit there. So this already, from the several axioms, is coded by a certain harmony between, after all, there are these universal rules that say, all I got to determine what I'm going to do next are my spectrum and the received signal. Therefore, if I'm instructed to do nothing, there must be a certain harmony between my spectrum and the received signal, which tells me to do nothing. So as a consequence, 1 through 4 already imply this interesting thing, and then I'll draw a picture, and you'll see it. There's somehow interesting content to this. 1 through 4 imply that an adult cell decides, let me put it that way, decides to be in equilibrium via a harmony, and again, this concept will return later in the lecture, a harmony between spectrum and received signal. Spectrum and received signal. So let me just draw sort of an ansatz experiment kind of example. Imagine an adult organism that looks like this, and imagine coming with a knife. So everything's in harmony. The cell here is receiving from all its neighbors the instruction, sit there, be happy, and you're in equilibrium. And now imagine excising this. That will alter the signal of the neighboring cells, including, possibly this neighboring cells, and you will no longer be in harmony. So I began the lecture by saying, where is this information coded? Well, the shape of an elephant or the shape of a giraffe, and it is coded precisely coded precisely in the harmony between my spectrum and the spectra of my neighbors. It's kind of, I think, kind of a beautiful, and this answers a question already. This is interesting. Where is the information coded? It's coded in this harmony, this notion of harmony. Okay, so we've already got some pay dirt, is what I'm trying to say. Or it feels like we already have some pay dirt here. Right. So let me continue. Now with some discussion. Oh, I guess we can go straight to an axiom. And this is interesting. So 5. There are optimal sequences, and I want to emphasize sequences, for we have experimental evidence towards that end. There are optimal sequences of cell events driving the zygote towards adulthood, towards its adult shape, towards its harmonious, I'll use that word now, harmonious adult shape. So adult shape. And this being sort of an example of one of these universal this being an example maybe of one of these universal rules. There are optimal sequences. Now I write the plural because we have experimental evidence that, I mean, you can go look in the literature at developing zygotes, and you can take the same species and follow the path of, say, the first several divisions of cell, first mitotic divisions, and they differ. So in other words, this is not a priori completely determined. It is not clear whether the plural here should be interpreted as a collection of discrete possibilities or maybe a distribution of possibilities. I think that this remains to be seen. But there are optimal sequences of cell events driving the zygote towards its harmonious adult shape. And maybe I want to say a little more that in particular, so let me elaborate. In particular, the collection of received signals, in the sense of two, the collection of received signals are somehow summarized into an instruction, and in particular a so-called target spectrum. So among the various data that are given to the cell, its position, its shape, and all this stuff, there is also this epigenetic spectrum. And part of the data, at any step, the cell is provided with a summary of its received signals, and I want to introduce the term target spectrum. In particular, a collection of received signals leads to, in some way that I'm not going to specify here. In implementation, you could choose proximity, spatial proximity. Maybe it has vectorial characteristic. I'm not going to try and be precise here anyway in the axioms about the nature of this target the nature of how you determine the so-called target spectrum. I said optimal sequences and target spectrum. Bang. And under normal conditions, the cell is provided with its target spectrum, and that is, say in embryogenesis, that is the next spectrum that it achieves. So there's some method of summarizing the signals, and the signals tell the cell to achieve a particular spectrum. And again, we can't quite think of it as unique. Maybe it's a distribution or something, or maybe it's several equally likely target spectra. But here, an example of I just wanted to give an example of one of these universal rules. One of these universal rules is that in normal embryogenesis, the cell moves temporally. The next state of the cell is the target spectrum, which is this summary of the signals it receives. And it just carries on in that way. Yes, sir? It's a word. No, it's a word. I mean, I don't have an answer in energy. Current work is attempts to formulate things in a kind of a Lagrangian sense, in the sense of physics as a Lagrangian. But let me not get into that. Optimal with respect to something that I'm not obligated to tell you now. We can have different models, anyway. This is to be probed. And I'll come at the end to how this model is to be probed. I mean, there are two aspects. One is computation, in silico, and another is in the laboratory, right? So optimal with respect to, I don't know. That's the answer to your question. But I wanted to emphasize sequences as opposed to sequence. Okay, so.
So in a sense, you can abstract it to say if a cell is a point, a vertex, then if a communication is an edge, then you could, and then what we're talking about in the state is a cell surface.
Cell surface distribution and a position and blah, blah, blah.
Then we would have a discrete dynamical system.
Absolutely.
And so in that sense, would you allow that particular graph or complex to evolve?
Yes, there are several graphs floating around here. Let me explain them to you. One of them is just the so-called adjacency graph. So as we've agreed, as I just erased, each cell has a well-defined point, namely the MTOC. So we have, for an organism comprised of its cells at any instant in time, we have a bunch of points in space, a few trillion for each adult in the room, right? I mean, the graph with a ton of vertices. Whenever two cells are adjacent, let us put an edge between the two vertices corresponding to the MTOCs of those cells. And in fact, this is not just a topological graph, but it's a metrized graph because there is a distance between the two MTOCs. This is not a completely arbitrary graph. For example, two cells can overlap. So there are constraints already on this adjacency graph. And that is how we think of it, that there is at any instant an adjacency graph with a certain number of cells, and at the next instant it has to evolve governed by some sort of Lagrangian or again, this is sort of current work, let me not go into that, and prolongs to the next adjacency graph and prolongs to the next adjacency graph and so on. And that is how we are thinking of morphogenesis in this context.
Because it would give rise to the idea that your harmonies may be even periodic points or...
Oh, who knows? Yeah, no, absolutely, absolutely. And maybe it's worth just mentioning another aspect that I guess I will mention. I wasn't going to mention that I will mention is once armed with the adjacency graph, there is a notion of distance between abstract metric spaces called the Hausdorff distance. So suddenly, by the time I'm armed with this adjacency graph, I can talk about the distance between a giraffe and an elephant. And this is a very useful notion, right? So in some sense, this star convexity and the shape of a cell allowed us to compare shapes of cells on the microscale. And the Hausdorff distance between the two metrized adjacency graphs gives us a useful measure of large-scale evaluation of distances. And maybe now let me elaborate a little more. I'm not sure I'm doing for time. I have a little enough time. We have 2 more axioms, by the way. So I have a moment. Speaking of René Thom, who wrote extensively about morphogenesis and was troubled with the following problem. How do I come up with a notion of distance between organisms after organisms move? You know, how am I going to tell the difference between me, and me, or whatever, having sitting down or whatever? And in fact, the Hausdorff distance between the adjacency graph. Hausdorff distance is well known in the computer science community to allow exactly comparison of shapes that are moving. So this conundrum of René Thom is really solved by this little discussion we just had with adjacency graph and Hausdorff distance. And there's a certain irony here because Hausdorff distance, the rich cousin of Hausdorff distance is Gromov-Hausdorff distance, where one compares not just metric spaces, but metric spaces, measured metric spaces. Each metric space comes equipped with a measure. And the measure for us could reflect internal cell contents too. I just want to point out our model is sort of more flexible and could allow also internal cell protein gradients. And I don't know what, whatever you want. And there's a certain irony that here's René Thom puzzling over this question, and down the hallway is Misha Gromov, and they could have resolved this themselves. But this was a puzzle, and I think I just gave a solution to something that troubled René Thom deeply about shapes of organisms. You satisfied with that? Okay. All right, we come to the last, saving the best for last. We come to the last 2 axioms. And the next one is kind of fascinating and pertains most specifically to regeneration or transplantation or some sort of chemical intervention of I'm now going to jump from normal embryogenesis to various crises of transplantation or whatever. And I say if the optimal cell event, so again we have this notion of optimality, some notion of optimality, if the optimal cell event is for some reason unachievable, event is for some reason unachievable, A-B-L-E, unachievable, then the cell has very few options. It can either do nothing, it can stagnate, it can die, then the cell either apoptosis, dies, stagnates, just sits there and waits for new signals to instruct it to a new optimal cell event, or perhaps the most interesting of this trichotomy, or de-differentiates. De-differentiates. By which I mean, and again, so here's the definition, its spectrum returns to that of its mother. Its spectrum, epigenetic spectrum in the sense of one, returns to that of its mother, a kind of a backtracking of normal morphogenesis to that of its mother. And let me just, you know what, I should have said something else about one. Of course, the zygote inherits its cell surface distribution from the mother, comes equipped with a cell surface distribution. That's sort of the initial condition for the entire thing. I should have said that. And now I want to say that this cell surface code is evidently highly redundant. And that is what allows this de-differentiation. For, imagine a cell undergoing mitosis. So it sort of pinches off with its neck, some band pinching off, and then ends up as the two daughter cells. And of course, this, I'm arguing, is covered with its cell surface distribution of oligos or whatever you want. And it's perfectly reasonable and quite compelling to assume that somehow this part of the cell surface is simply, the cell surface distribution is just inherited by the daughter. I mean, this is happening continuously in time. And so clearly, part of the cell surface distribution of the daughter is just picked up directly from the mother. And one of these universal rules tells us how to fill in the rest of the daughter's cell surface distribution. That is among these universal rules that I'm, again, not elaborating. And so there is this great redundancy. And so there's the potential for great redundancy in the epigenetic spectrum, which permits this de-differentiation. And so you want to imagine if a cell can't do what it's instructed to do, it takes a step back. And now if it still can't do what it is now instructed to do, it takes a step back, and so on and so on. And this is exactly what happens in a blastema. There is this proliferation of cells, and de-differentiation really explains and provides a dynamic for that. So, like I say, saving the best for last, I think. The most interesting, the most mathematically interesting axiom, I think, is this one, and it takes a little bit of time to explain. And is as follows, and we'll return to the blastema armed with this in a second. I have yet to explain the dynamic. So now you want to put this on the computer and you want to set it off and something happens and there has to be a dynamic. And here, let me explain the dynamic. Namely, the strength of the sent signal of a cell to its neighbors, in this generalized sense, varies. I think this is the most beautiful. Anyway, this was really the last piece of the puzzle, as far as I was concerned. The sent signal varies inversely as its potency. And yeah, I haven't defined potency, and now I will try and do so. The potency of a cell is qualitatively its ability to produce many different cell types of progeny. So, for example, the zygote is totipotent, produces everything. And indeed, many of the first cells post-mitosis are also totipotent, according to many experiments. One imagines, it's kind of like entropy. One imagines as the zygote evolves, the potency of the cells diminishes. Finally, in an adult cell, say an island, the cell of an island, it's zero potency. It's just, there it is. In the case of say, a liver cell, or an adult stem cell, it's bipotent. Namely, it produces either the differentiated cell type, it produces the differentiated cell type and another stem cell to do the same. So liver cells and so on, many examples of that. I'm just trying to give a qualitative idea. I think everyone in the room knows what I mean by potency. We have spent much effort to try and give an explicit either mathematical or biological definition of potency without success. Once you postulate a sequence of an optimal sequence, it's easy to define the potency of a cell. You just look at all the downstream stuff and count out, in some measure, how many there are, right? That's easy. But absent a particular sequence of cell events, how can I possibly define this notion of potency? Well, I could take the cell and let it evolve under all possible laboratory conditions and see what comes. That's absurd. That's completely unmeasurable. You know, forget it. Or I could take the cell and put it in particular laboratory conditions in a plate and blah, 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 at a particular temperature and all that, and see what evolves. And that's too specialized. We have really spent much effort to try and give an abstract, either biological or mathematical definition of potency that's intrinsic somehow to a cell, and have failed. So I don't quite know how to formulate that, except in its relationship to an optimal sequence of cell events. So let me show you why I think 7 is so beautiful. Imagine, so among the, let me move this up, I suppose. So again, in the literature, there are many examples of bizarre experiments of transplantation. And you cut off an axolotl's leg and it grows a new leg. You cut off an insect's leg and turn it 180 degrees and glue it back and it grows 2 legs. There's an organism, planaria, a worm, that basically you take any fraction of the worm and it grows back it reproduces in nature in one of 2 ways, either sexually or it just rips itself in half and grows a new head and a new tail. But in fact, the planaria are filled with stem cells. And in fact, you take various segments and it reproduces a new organism. And there are many beautiful and weird experiments. If you put so cut the planaria, cut off the head and the tail, put a voltage across it, and it'll grow 2 heads. Unless you put too much of a voltage differential, and then it grows, then it does it normally. They're bizarre phenomena. But anyway, I wanted to explain the dynamic of sort of 6 and 7 together in terms of these regeneration experiments. Okay, so here's this poor axolotl's hand that we're going to chop off. And imagine, you know, cutting here and producing this stub. You know, in practice in these experiments, like I said, a blastema forms, namely sort of a bubble of undifferentiated, a profusion of undifferentiated cells, a so-called blastema. And there is this wave, first of proliferation and then of differentiation. And so the proliferation and stemminess, high potency, is explained by this dedifferentiation. These cells are just sitting there as they were, probably in harmony with the nearby. I'm returning to the notion of harmony and explaining how harmony drives regeneration. So down here, the signal presumably is the same as it was. This guy is in stasis. This cell can't do what it's supposed to. According to Axiom 6, it dedifferentiates, and that explains the evolution of the blastema. Of course, there's tendons and there's muscle and there's bones, all these differentiated pieces that are still sitting there. And now, according to 7, the bone is screaming, bone, 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 at a loud volume, and the tendon is screaming, tendon, tendon, tendon. And these stem cells are, stem. And so the received signal, this explains how the harmony of signal also explains regeneration in these sorts of experiments. So this is the role of this inverse potency law 7. Again, these are screaming, these are silent, so this cell is receiving a signal to be a bone, 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 bone. And then there's this wave of differentiation and presumably further blastema, further proliferation of stemminess, and blah, blah, blah. And you can see how this kind of wave of differentiation extends up here and reproduces the leg. I think that's a very beautiful dynamic. And look, there's this basic question I think we've answered. where is the information stored? And these 7 axioms together explain many experiments in the literature, including in many aspects of embryogenesis. So let me close with what we are doing here. Let me aggrandize everything by putting it all at once. And explain that we have there's a paper that's in process and nearly finished where we implement this on the computer. So obviously now you start probing this in silico and see what happens. And in fact, we have what we've done, it's sort of artificial, but what we've done is taken random initial epigenetic spectra on the zygote and just let her rip, make some assumptions like there are all sorts of parameters here, but I haven't specified. So make some assumptions about that, like division planes are always in the coordinate, in the embryonic coordinate axes or something, blah, blah, blah. You make some decisions how to implement these, and then let her rip and see what you get. And we get, for certain of these random distributions with our eyeball, spectacular agreement with actual embryos. So these virtual embryos, we're able to produce virtual embryos that look to your eyeball very much like actual embryos with ridges and so on. I mean, it's striking. It's a little bogus because we're cherry picking, right? We're taking random distributions, letting it rip and saying, I like that one, I like that one, I like that one. Look at how well they fit. So it's a little bogus, and in fact, it's also a little bogus because I said with my eyeball, we're also in the process of implementing this Hausdorff distance to actually compare embryos. And that's what's holding up the paper a little bit, is we're in the process of implementing that. So there's the in silico probing of this, and obviously we need to do some experiments. So and there are methods of tagging oligos in the cell surface, and unfortunately there are only so many channels you can do at once. So in practice, we want 12 or 15 or something species. We're going to have to take several zygotes, and then we're sort of plagued with the fact that this was a plural. The optimal sequences, I mean, we're plagued with that fact. But the bottom line is that we need €1 million to buy the equipment to do these experiments, which maybe seems like a lot or maybe doesn't. But I'm happy to take donations towards this end. Anyone who wants? Or perhaps, Chris, there is some wet lab here that might be interested in collaborating. Anyway, here's this model. We're probing it in various manners, and the next step is sort of wet lab probing of it, and hopefully that will be not in the too distant future. In the not too distant future. So thank you. Questions? Yeah. Yes. That is the presumption. And again, I think of it as a sort of an entropy.
Is that encoded axiomatically in your system? Does it fall out anywhere?
No. No. And what I said is wrong. Well, under normal evolution of a zygote, the presumption is yes. But look, this example of the blastema and the dedifferentiation, so no, it's not universally true. In the case of crisis, something can dedifferentiate, return to its mother, namely increase its potency. But these are only, the normal evolution of a cell is to decrease potency. Yes, sir. Roughly, yeah, no, absolutely, irreversible. And that's why I drew this picture, is that if there was, say, a twofold symmetry in the mother, then the daughter can just return, returns to the mother's epigenetic spectrum. This picture was meant to argue there is we presume there is high redundancy in the epigenetic spectrum, and that's why the daughter can return to the mother, and even the grandmother, and even the great-grandmother. Yes. No, but it doesn't return to its. Now it will have new signals. See, in the case of crisis, now it will have new signals and a new target spectrum and a new evolution.
So it won't go back.
It goes back, but I don't know what you mean by back. It goes back in sort of the tree of cell events, but then doesn't return to where it came from because now it will have new signals. It will have a new signal, and the universal rule will tell it, no, no, don't do that. You know, move over here, take this shape, evolve this way. Right? You see?
So I have a question, but I have to run out for a minute.
Sure. Go ahead, Chris.
I can ask it before I run out. And then, as long as you want.
You the boss.
Because it's your lunch.
Okay. No, we can stop it.
So this is the question. So I sort of see the picture of this. I really do want to see what the mathematics of talking about this carefully are. But in number one, it seems like there's a 0 that isn't on there, right? And it seems like the 0 is something as follows, which almost makes some sort of a continuity sort of thing here. And that is that there has to be, you know, like this semiotic thing, signs and signals and stuff. These compounds that you're having in the spectrum, they have to be made by the cell. They have to be made and delivered to this, which means they're actually part of the cell. Now,
True.
they're part of the epigenetic environment for the other cells, but they are a sign of, they're correlated with whatever it is that is specific about this cell, right?
Yes.
Which means their latent existence is present in whatever the code is that produced it in the first place, and which brings you back sort of over to here, you see, and
Over to
7. And so that the
Sure.
right, and so my what I'm sort of want to just ask is, these compounds are not unrelated to the cell itself. They're part of the cell itself. Right?
Surely.
And they're produced by it. They're part of the definition of it. They're sort of like the name of it painted on the outside of it or something. But does that, first of all, is that even sort of right?
No, that's all.
And secondly, does it change how you think about this if you have to include that?
I don't see a change in including that. Those are, so first of all, like I said, the zygote inherits a very elaborate oligocode from the ovum. Comes equipped with something very elaborate. So first of all, that's the initial condition. And I think I would argue that it is the universal rules that tell you how you fill in and continue. There's some universal rules that tell you how to complete the code. And again, I was trying to argue here that most of the code, or much of the code, or half of the code, make some odds, half of the code is just straight up inherited, and then there's a universal rule. This is now embedded in 4. There's a universal rule that tells you how to extend.
Okay, well, I hope I can talk.
Yeah, no, I'd like
I can
that.
talk to you about it a little later, but
Sure,
I have
you
to
bet.
go right now.
Okay, you got to go cut a ribbon. And perhaps, unless there's another pressing question, we should wrap it up because I think there are constraints.
Go out on a limb and thank the morning speakers in totality, and it's time for lunch. Thanks.
My pleasure. Thank you.
And don't forget to come back for the Hautes.
Ah, yes. Okay. What time do we proceed here? 1. You want to say it out loud or something?